All right, here I'm going to describe a biochemical pathway for you. We're going to start with the concept that we have two different types of pathways. This isn't always correct, but this is the easiest one. This is the one that you are most likely to hear about. And the two types of pathways are catabolic, all right, and anabolic. A little longer there. All right. Now, the one that you might have heard of more likely, or at least more likely to have heard of, is the anabolic, like you might have heard of anabolic steroids. And what this means is to build up, all right, or put together. All right. Catabolic, all right, unlike the anabolic, means to break down. So, what we're saying here is that whatever this thing we're calling a pathway is, that one of them kind of tears things apart and the other one puts things together. The anabolic, steroids, you grow when you take steroids, anabolic steroids, like bodybuilders, weightlifters, they're often illegal if you're an athlete, but if you've just had surgery or something, especially on the brain, where you need to heal that wound very quickly and close it up so that you don't get an infection in your brain. So if you've had like brain surgery, they'll give you anabolic steroids to make sure that you heal and you close a wound absolutely as fast as possible. So that you would want to build up. You're building something inside your body, so you would want to take anabolic steroids. Catabolic is what happens if you starve yourself. Your body will go into catabolism and start breaking itself down to produce energy. You don't eat for a few days, well, your cells will activate catabolic pathways, and they will start breaking down the parts of the cell to produce energy. If you have plenty of food, and you're more likely to have anabolic pathways activated. So these are pathways, and I have to explain to you what these two pathways are, or at least some semblance of how they work and how they're related to genes. So let's get a new blank image here. Let's expand it a little. Oops, wrong way. There we go. All right, so let's start with a catabolic pathway. Now, here we're not breaking down yourself. You're not starving yourself. But let's say you've eaten some food, and you have some food here. All right, and that food, you want to digest it. Now, you can't eat the food in the form that it's in chemically. So here your food, write that up here, is not digestible in this way. Instead, you need to break it down chemically into smaller components, and then these need to be broken down into even smaller components. And that would be chemically, not this is just size, and then all of this into the smallest components. So maybe you ate something that complex carbohydrate, and if you know anything about carbohydrates, they have lots of sugars essentially bound together. But that sugar isn't useful to you. You need glucose. So you would have to break down this complex carbohydrate into a simple sugar like glucose. And the idea is, how do you do that? Well, you do this using different genes. You have enzymes, proteins, that are produced by these genes. So going from here to here, somewhere in between there, you have to add to this food a chemical, a protein called an enzyme that will catalyze, that means speed up a reaction. The food might have broken down by itself if you just let it sitting out, but you want to break it down now. You want to add something acidic, or you want to use a protein that can go in and literally chop this food up, these carbohydrates up into smaller components. Well, you would need a particular enzyme. So this would be an enzyme is a type of protein usually. So this would be protein 1. All right. And then here, going from this to here, once you've produced this particular substance, you would do protein 2. And then to go finally from here to here, you would use protein 3. So you need 3 proteins. Your body needs to produce these 3 chemicals, these 3 proteins, in order to go from the original food you ate down to the basic glucose that you want. So this is like pasta or something, and you break it down and you make it simpler. You break it into simpler components and then simpler, and finally you get to glucose down here at the bottom perhaps. Well, what produces these proteins? Well, the proteins are produced by genes. The genes code for their instructions for how to make these proteins. And so each one of these corresponds to a gene. All right, so let's say on a chromosome, I have up here a piece of DNA that codes and it's the instructions for protein 1. So we'll just call this gene 1. Oops. Running out of room. So here is gene 1. Then a little further down here is gene 2. And then here is gene 3. All right. And so each of these genes, they're sequences of DNA that are basically able to produce an RNA. And the sequence of the nucleotides, the A, G, C's, and T's here, are instructions. They're instructions telling a ribosome how to produce a protein. So here, gene 1 tells the ribosome how to produce this protein. Gene 2 tells the ribosome how to produce this protein. Gene 3 tells it how to produce the third one. And the idea is, in the case of all 3 of these, all right, they work together. They have to be produced in certain amounts at certain times. So it doesn't make sense to produce this one when you just ate this one. You need to produce this one first. Then you need to produce this one here, P2, and then you need to produce P3. So there's a timing that's involved, and there is an order so that you do this one first when you eat food. Also, there has to be something to activate the production of this protein. So some environmental factor, which is the presence of this food. So you eat this food, and that's an environmental effect that turns this gene on. At the very least, it makes you produce more of it. If the gene is activated, you may not be producing much of it at any given time, but an environmental factor will cause you usually to start producing this. In this case, you ate food and it's in your stomach, or maybe it's smell. It can be all kinds of interesting things that can actually activate and turn this gene on or upregulate this gene. Well, this gene is the first in a pathway. So our pathway is going from P1, then to P2, and then to P3. These 3 genes, or alternatively, these 3 proteins, you can view it either way, are together produce a pathway. But you have to understand that there's an order and a timing about it. There's something that activates them, and then they have to be these proteins have to be produced by a particular type of cell. They may not be the same types of cells. For instance, this one might be produced in the stomach, this one might be in the small intestine, and this might be in the large intestine. So that means different cell types might have to produce these, but as a general rule, chemically, if I were to take this food outside of the body and add this protein, all right, it would break it down here. And then if I add this protein artificially outside of the body, it would break it down like this. And then finally the third would break it down this way. And if you produce the same chemical conditions and add these proteins, essentially you would get the same thing. And so we know how the chemistry of these proteins work. And when we find how these proteins and genes associated with them, we call that a biochemical pathway. Now, how does that relate to us in our problem? Well, in our problem, we're looking at genes, and we're looking at genes that produce the same phenotype. Well, what if you have a disruption? All right, let's see if I can make this happen. And something causes protein 1 to not work. So protein 1 stops working. Well, if protein 1 stops working, you basically don't get glucose. All right, and that's a phenotype. You can't digest glucose. You can't eat food and you can't eat carbohydrates and get glucose from it. That would be a mutation. You would have an allele in this particular gene where it didn't work, it didn't do its job, and therefore you can't produce glucose from this particular type of food. But you could get the same phenotype, all right, if what? If you just had a mutation here. So this one might be okay, protein 1 might be okay, but you could get the same result if you mutate protein 2. So protein 2, if you get an allele here, basically you can break it down to here, but you can't keep going. And so both of them produce the same phenotypic result, the inability to break this particular substance down into glucose. So since you cannot break your food, your pasta, into glucose, you have this particular phenotype. This is something that is expressed in you as an organism, say. And if you get a mutation in any one of these three proteins or in these three genes that disrupts its function, all right, then you're going to produce the same phenotype. So this is how you can have one phenotype produced by mutations in any one of these genes. So if you have mutations that produce an alternate allele that leads to non-function of any of these genes, in other words, they don't produce the protein correctly, so they don't chemically function correctly, if you have mutation in any one of these, they would all produce the same phenotype, but they would all be different alleles on different genes. All right. Now you might have different alleles, different ways of disrupting protein 1 or protein 2 or 3. So there might be multiple alleles that also produce the same phenotype. In other words, some mutation, there might be more than one mutation that will cause protein 1 to not work. And probably thousands of mutations you could have. Depends on how many letters you have in your in the gene, how many nucleotides are in that gene. Any one of those changes might alter the functioning of this protein. So any one of those mutations might lead to it to not work, and therefore you would be producing a different allele. Every one of those different mutations. Now that can't always be identified. They might be several mutations, and we may have only identified a few, but now that we can sequence genes, we go in and we can see at the DNA level how the mutations produce the alleles. And so the changes in the letters along the chromosome that make up this gene, here, the nucleotides, we look at it and you disrupt some of the letters and suddenly this doesn't work. Now we would in standard genetics, we would call that, all of these would be the same allele. But as we learn more, and we learn more in the bioinformatics side of things, and we call it molecular genetics, we begin to realize that, okay, each one of those is actually technically a different allele. It's a different mutation. Now, it just happens to produce the same product, just like mutations in any one of these might produce the same phenotype. We have mutations here in the gene which might produce the same allelic change and produce the same, as far as we can tell from the classical genetic level, the same mutation, but it actually came from different mutations along the gene. So this is the idea behind how you go from mutation in a gene to allelic, the concepts of an allele, finally to the concepts of a change in genotype, and finally phenotype. So realize today we understand classical genetics is really all based on changes in the nucleotide sequence, and which lead to changes in the genotype, which then of course change the phenotype. But we used to not have this gene component, and only now have we learned how to even manipulate it so that we can even repair these at times with something called CRISPR. All right, that's it. Hopefully that helped.